It's the tiniest tape recorder in the world, and could open the door for a massive change in everything from disease diagnosis to environmental monitoring. Researchers at Columbia University Medical Center have converted a natural bacterial immune system into a microscopic data recorder. By modifying a strain of the ubiquitous human gut microbe E.coli, they enabled it to not only record their interactions with the environment, but also timestamp the events. Such a bacteria, swallowed by a patient, might be able to record the changes they experience through the whole digestive tract, yielding an unprecedented view of previously inaccessible phenomena, says Harris Wang of CUMC, the senior author of the science paper. Other applications could include environmental sensing, and basic studies in ecology and microbiology, where bacteria could monitor otherwise invisible changes without disrupting their surroundings. The researchers created the microscopic data recorder by using CRISPR-Cas, an immune system in many species of bacteria. CRISPR-Cas copies snippets of DNA from invading viruses, so that subsequent generations of bacteria can repel these pathogens more effectively. As a result, the CRISPR locus of the bacterial genome accumulates a chronological record of the bacterial viruses that it and its ancestors have survived. When those same viruses try to infect again, the CRISPR-Cas system can recognize and eliminate them. The CRISPR-Cas system is a natural biological memory device, says Wang. From an engineering perspective, that's actually quite nice, because it's already a system that has been honed through evolution to be really great at storing information. Over a dozen clinical trials are now underway to treat various diseases through CRISPR-Cas gene therapy. But Ravi Sheth, a graduate student in Wang's laboratory, saw an unrealized potential in CRISPR-Cas recording function. When you think about recording temporarily changing signals with electronics, or an audio recording, that's a very powerful technology, but we were thinking, how can you scale this to living cells themselves, says Sheth. To build a microscopic recorder, Sheth and other members of the Wang lab modified a piece of DNA called a plasmid, giving it the ability to create more copies of itself in the bacterial cell in response to an external signal. A separate recording plasmid, which drives the recorder and marks time, expresses components of the CRISPR-Cas system. In the absence of an external signal, only the recording plasmid is active, and the cell adds copies of a spacer sequence to the CRISPR locus in its genome. When an external signal is detected by the cell, the other plasmid is also activated, leading to insertion of its sequences instead. The result is a mixture of background sequences that record time and signal sequences that change depending on the cell's environment. The researchers can then examine the bacterial CRISPR locus and use computational tools to read the recording and its timing. The current paper proves the system can handle at least 3 simultaneous signals and record for days. Now we're planning to look at various markers that might be altered under changes in natural or disease states in the gastrointestinal system or elsewhere, says Dr. Wang. Synthetic biologists have previously used CRISPR to store poems, books, and images in DNA, but this is the first time CRISPR has been used to record cellular activity and the timing of those events.